Yeah, hi everyone. I'm Oliver. I'm not Chong, I'm a different person. I'm doing a PhD at the ANU with Sasha Mikheyev, collaborating with people down in Melbourne, actually, at the Melbourne Zoo with Zoos Victoria. And all of my PhD projects are kind of proceeding in tandem. So I'm going to give you something of a whirlwind tour of a couple of different projects that are going on with D. Australis, the Lordhow Island stick insect. So D. Australis is a critically endangered insect. It used to be abundant on Lordhow Island in the South Pacific, which is an Australian territory now. And then it was very quickly driven extinct by rats in the early 20th century. Following that, it was presumed globally extinct for about 80 years. And then in 2001, was rediscovered on Ball's Pyramid, which is about 25 kilometers to the southeast. At present day, this population is still kicking around, so not extinct yet. And we now have a large captive breeding program operating at the Melbourne Zoo. So because the extant wild population is extremely small and also has an extremely restricted range, it's definitionally very, very critically endangered. Now, to my mind, the questions that come to my mind that are most interesting about this species are mostly population genetic questions, like things about deleterious substitution rate, heterozygosity, founding events, those kinds of things. And those have turned out to be extremely difficult to get clear answers on. And so a lot of what I've been doing for my PhD project has been answering much more basic questions about the species genetics. So first thing I'm going to talk about is exactly what the D. Australis genome actually looks like. Ooh, where am I? There we go. Jumping right into it, I can tell you that the D. Australis genome has a diploid karyotype of 34 chromosomes and a parent XY sex determination. The reason that we are pretty sure it's XY sex determination is because we have two different individuals on screen, and on the left, there are two heteropignotic chromosomes, heteropignotic, that is to say, staining more darkly. So they have denser chromatin than the others. So on the left, two heteropignotic chromosomes that are the same size. And here we have two heteropignotic chromosomes that are different sizes. Heteropignotic chromosomes are very often the sex chromosomes. And when we have one individual which has a homokaryotype and the other which has a heterokaryotype for these, we assume that those are X and Y. So the curious thing about this is that on the picture on the right, the larger putative sex chromosome is probably the Y chromosome, and it's much larger than the putative X chromosome. Y chromosomes are usuly quite degenerate. The heterokaryotype sex usually has a degenerate chromosome across all chromosomally sex-determined species. And so we think it's probably quite a young Y chromosome. To this end, we've also obtained a very high quality genome assembly, 99.5% of which has been assembled into 17 chromosomal scaffolds using high-C data. 17 chromosomal scaffolds agrees very well with that karyotype estimate of 34. But despite the high contiguity and structural agreement with our karyotype in this assembly, we're still missing about 30% of the assembly's total length. So we estimated genome size with flow cytometry at about 4.8 gigabases, so 4.8 billion bases. And our genome assembly is only 3.4 to 3.5 billion bases. And we're pretty sure that this high missingness is due to the high repeat content of the genome assembly. So the figure that you're looking at is just the proportion of sequence that is masked by a basic repeat masker pipeline across the length of all 17 major scaffolds in our assembly. And it sits at about 60 to 80%. So this is a large, highly repetitive genome, which is very difficult to assemble, even with quite dense long read data. Not ultra long, but long read. So the next interesting thing about this assembly and about the species genome is we have this one, spoilers, we have this one scaffold creatively named Scaffold Fork, it's the fourth largest, that retains 60% of the X chromosome content from Tomema. So Tomema is the outgroup genus of stick insects, the most early diverging genus. So we're pretty sure that this is a sex chromosome scaffold, probably the X chromosome. What's remarkable is that all of this X chromosomal gene content is retained despite between 120 and 300 million years of divergence since the last common ancestor of stick insects. And just to complete the story, this same scaffold that has all of these X chromosome genes also has half depth in a male long-read data set compared to a female long-read data set. And this is consistent with 2 X's in a female and 1 X in a male. And just taking everything together that I've said so far, what we have at this point is a very high quality genome assembly, structurally similar to our karyotype, which is very nice to see. And what we are pretty sure is a very long contiguous X chromosome assembly, which will be very useful for future comparative genomic work with other stick insect assemblies. Now, this is a karyotypic observation that I, as yet, cannot explain. So this is a male meiosis in D. Australis. So I got testis samples from a number of males from the captive population. And when we karyotype those, we can not only look at mitosis, we can look at the full chromosomal complement in somatic tissue. We can also look at meiosis. We can look at how the species produces gametes. And in all of the males that I investigated, I saw what appeared to be 6 bivalents segregating at meiosis. And try as I might, I can't make the number 6 go into 17 or 34. So there are 2 possible explanations for this. One is that in all of the males that I investigated, meiosis is somehow aberrant, which would obviously be of interest to the conservation program trying to keep the species breeding. Or there is something unusual about meiosis in this species. Unusual meioses and unusual meiotic pathways are not at all unheard of in invertebrates, especially Australian invertebrates, where this is very reasonably well researched. So that brings me to the next basic biological question, which I'm looking at, which is how does inheritance proceed in D. Australis? I don't yet have a heap of data on this, so I'm just going to talk you through the study design so that you can understand what kind of information we're getting at. Currently at the zoo in Melbourne, the zookeeping team are maintaining 12 controlled crosses between 2 distinct genetic lines. So they've got a number of different genetic lines going in the population, managed in slightly different ways. One of these is the LHIP line, which is partially descended from a recently caught individual, recently caught from the wild. And so these individuals have spent 3 to 4 generations in captivity. And the other is the LHISI line, which is a generationally discrete line, which has been descended from the original founders of the population. So they've spent about 16 generations in captivity. And we have reciprocal male-female crosses going of both, so the zookeepers can get an idea of what the effect on fecundity is of crossing distinct lines. But what I'm doing is I'm collecting the eggs from these crosses, dissecting out the embryos, and then at the end of the adults' lives, sampling tissue from them. And what we'll end up with at the end is a large genotype data set with shallow pedigree information, so offspring matched to their parents. And the point of this is threefold. We want to construct a linkage map for the species. We want to validate a set of markers for ongoing monitoring, so markers that are segregating in a decidedly Mendelian fashion. And we also want to look at patterns of segregation from parents to offspring in light of the strange meiosis, which I showed you before. And for purposes of the next figure, I just want you to think of these 2 lines as crosses that involve outbred males and crosses that involve inbred males, rather than trying to remember the names of the lines and what they mean. Because the last thing I'm going to talk to you about is how D. Australis may have adapted to captivity. So in the crosses where the male is outbred and the female is inbred, what we're seeing so far is that there are some apparent differences in life history. So on the top figure, you can see that in the outbred male crosses, the female started laying eggs slightly later on average than crosses involving inbred males and outbred females. And the second figure shows you that in those same crosses where the female started laying later, they also started producing viable eggs considerably later than when males were inbred and females were outbred. And this would be consistent with adaptation to captivity involving a shifting forward of the life history. So faster development, earlier adult fecundity, that sort of thing. And this also agrees also agrees with some early phenotypic data, which I'm compiling from zoo records stretching back to 2003. This is the days to hatching of eggs in the first generation in captivity. So the eggs laid by the first female, very creatively named Eve, and eggs laid in one of the recent generations, generation 16. And there has been a shift forward in the hatching time in this particular line of the glasshouse. And that's pretty much everything I have for you. So I'd like to thank Dave Roll at the ANU, who gave me a lot of help with karyotyping, the zookeeping staff down at the zoo, who put in a monumental amount of effort maintaining these insects, Darren Parker at Lausanne, who provided me with a list of X-linked genes for Tamima, funding bodies, the Holsworth Endowment, Dovetail Genomics, Zoos Victoria, and of course the Entomological Society for giving me the opportunity to speak to you today. That's all I got. I'd love to take questions. Thank you very much.
Very much, Ollie. Very interesting stuff. We have some questions here. So Ari's got a question. Ari, did you want to ask that?
Yeah, sure, Ollie. I was looking at your graph, and you only popped it up very quickly. The last one, I was trying to work out there's a shift in the variance, because it seems to me that the variance was also reducing potentially as a consequence of the lab adaptation. I'm just wondering if you've looked at that and whether you could comment on that, because it might indicate something about the nature of the selection going on.
Yeah, so I haven't yet looked at that in detail. I'm still combing through the records, but actually I'll see if I can just go back to it. Here we go. Yeah, so there does appear to be a reduction in the variance in hatching time. You are right. All I can say at this point is, yeah, that is an interesting point and something that will be really, really good to look at when the data is completed. So I will have data for 2003 up to present day completely compiled in the near future. That's a good question. Thank you.
Yep.
Anyone else got a question? I've got one for you, Ollie.
Yep.
So you've got this pretty slick genome. Maybe, you know, it does have a lot of repeats in it, but it does seem like it's pretty, you know, you do have like a full assembly, relatively speaking. So what are the challenges then associated with you performing these, you know, population genetic statistics that would be quite informative for conservation programs, given this excellent resource?
So the main problems we were having before were when we were trying to design markers without any decent genomic information. So a lot of the early analyses that I did with purely, so for instance, mapping back RNA-seq reads to an assembled transcriptome was one of the first things that I was doing. And I was finding a lot of off-balance SNP calls, even a lot of the on-balance SNP calls. So heterozygous SNP calls with an allele balance, you know, from 0.4 to 0.6, whatever threshold you want. They were still showing they were still showing extremely high heterozygosity across all individuals. We were seeing like negative FIS in pretty much every sample investigated, which is almost certainly artifactual in a species in captivity with such a small wild population size. And those have been mapping back those reads to the actual genome assembly. We're getting much fewer artifacts of like over-assembly and duplication. So having the long reads to assemble a more contiguous chromosome has really helped us, at the first pass at least, get over our SNP calling artifacts. I spent a good 6 months chasing bioinformatic ghosts related to like over-assembly and mapping to repeat regions. And it's obvious why that's the case now, because it's so repetitive.
Awesome. Thanks, Ollie. Really great talk.